Good morning, everyone. Nope, not good morning. Good afternoon, everybody. Hope you guys are having a good start to your summer. Hope that everyone had a good day off yesterday. Um, so this is Chem 4375. I'm going to navigate through our Canvas page a little bit, and I'm going to give everyone a couple of minutes or so to populate in. Um, We've got a grand total of 25 folks in this class. We've got 16 participants right now. I'm not going to wait for 20 minutes or anything like that, or even 5 minutes. Um, but I'm going to go ahead and stall for just a little bit. Okay, I've stalled long enough. So this is Chem 4375. This is more than likely your first biochemistry class. Um, if you are taking this class, that means that you are not a biochemistry major. That is one of the most important things that I kind of clear the air on. Um, Biochemistry majors take Chem 3375. If you are interested in being a biochemistry major, but you want to kind of test this class out, provided that you understand that this will not contribute towards your biochemistry degree, then I'm all for that. But I hope that everyone who is in this class has at least communicated with or knows that they are taking this class kind of intentionally. I know that people don't really sign up by accident, but it has happened. Folks who have taken this class, they tend to be biology majors. There's a handful of chemistry majors, and then even some agriculture majors. Um, so there's a whole different range of fields of study for people in this class. The prerequisites for this class, if you are not aware, um, Gen Chem 1, Gen Chem 2, Organic 1, and we have Organic 2, I'm sorry, 2342 and the accompanying lab listed as a co-requisite. That's kind of a relatively new change. And to be completely honest with you, I think that it's pretty reasonable. Just because if you're taking organic 2 or have taken organic 2, you know that organic 2 is lots of mechanisms. It's pushing electrons around. It's, oh, how is this bond made? Um, and whereas organic 1 is a bit more in the nomenclature and making sure that you understand fundamentals and establishing what those fundamentals are of like polarity versus non-polar and everything like that. So, in my opinion, if you've taken organic 1 and you've done passing in that, then I think that you can pass and do well in this class. Um, if you have not taken a biology class in a long time, that's fine. There is no biology prerequisite for this class. I think, however, it is important to recognize that this class is biochemistry. So if you're thinking, oh, well, I never want to learn anything about a living system, well, go ahead and put the brakes on that because that is kind of the area that we're living. This class will focus on kind of physiology and the relevance of a living system to chemistry. Okay, we're up to 21 participants. That's pretty good. Um, and so that's kind of the important parts about this in terms of prerequisite courses and prerequisite coursework. Now, what I want you guys to know are a couple of things, and I'm going to switch my screen. I hope that everyone was able to access the Canvas page all right, and I hope that everyone's taken a minute to kind of familiarize yourself with the syllabus. Um, I'm going to go ahead and jump over though, just to, make everything as clear as possible. Um, I will go ahead and first applaud myself for using a syllabus that has the correct date of summer 2025 as opposed to 2024 or spring 2025. And that's an indication that I did look at the syllabus and I did make the changes to make this appropriate and topical. Our class meetings, Monday through Friday, 2 p.m. to 3:40 p.m. The office hours that I will hold are going to be Monday, Tuesday, and Thursday from 12 p.m. to 1 p.m. Those are the two kind of most important things in terms of like where do you need to be and everything like that. Now, if you've looked at CatsWeb, and you absolutely had to in order to register for this class, you were probably prompted with something like, oh, this is an online class. This is an online slash hybrid class. So let me rephrase that. This is a hybrid class. Now, what does that mean? Well, we have at Texas State a couple of specific definitions. An online class is one where you will never meet in person. A in-person class, so opposite end of the spectrum, is where you will meet in person up to, or sorry, no less than 85% of the time. And so to kind of put actual numbers on that, an in-person class, your instructor has kind of a little bit of flexibility. They might have like 3 lectures in a semester that would be online, where you are not in the same location. A hybrid class is kind of the opposite. This might sound a little strange, but it's kind of the opposite of an in-person class. Because what that means is that we will be meeting online in 2 different locations, or at least 2 different locations. Now, let me rephrase that. We will be meeting in different locations online no more than 85% of the time. So let me rephrase that. What that means is essentially we're going to be meeting in person 3 or 4 times this semester. Now, I've elected to reserve those for exams. So exams will be administered in person. However, and I have this on the syllabus, if you are in a position where you need to take your exams online, I can accommodate that. I have no qualms with that. If you prefer taking your exams in person, they're available. And I'd just like you to kind of treat this as, hey, I need to contact me, and I don't need to know your complete life story or anything like that, but we might have a conversation and make sure that you're in a position that you have the kind of, the infrastructure in order to do that. Um, if you are one of the folks who will be doing that, you need to contact me before the exam date, so that I can make sure that you're all set up for that. Um, and we'll probably meet on Zoom and I'll discuss, hey, here's the technical requirements that are expected of you. Um, but if not, if you're going to be taking your exams in person, well, that's no problem. I believe that our classroom or the classroom reserved for us is Chem 100. Um, and so you just go there for the exam dates and your exams will be administered. Um, but that's that. Okay, so we got class meetings. There's your link. I've also added this link to our Canvas page. I thought that I did it whenever I posted the syllabus, but I did not. But click that link. You guys have all done this, and I don't see any emails from somebody who cannot join the meeting, so that's good. You guys all have your authenticated Zoom. Sorry, you are all authenticated users. Um, Office hours, 12 to 1, Monday, Tuesday, Thursday. And I just kind of spaced it out or separated it out a little bit just because, well, Monday seemed like a good day because those are when we're going to have our exams. Tuesday seemed like a good day because it might be, hey, I had my exam yesterday. I did as well as I expected. I did not do as well as I expected. What gives? We can talk about it pretty quickly thereafter. And then Thursday seemed like a good day to have office hours so that you could, hey, I've processed a little bit of this information. I'm having trouble with working on right now. Now, with all of this in mind, these are the designated times that I will be available on this link. So if you click that link between 12 and 1 p.m. on Monday, Tuesday, and Thursday, I will be there. Unless I send an announcement that says, hey, something unexpected, I can't make it. But I will be there. If you need to meet at a different time, if none of those times work and the best time for you to meet is Fridays at 8 a.m., then I need you to contact me. I have done this because in my years of teaching at Texas State and teaching in general, um, I've kind of gone over all over the map with office hours, been extremely rigid or been extremely flexible. And whenever I'm extremely flexible and I say, hey, office hours are whenever you contact me, well, then people don't use them. I've found that whenever I have designated office hours, I will be on this Zoom link. People will jump in for 5 minutes and then they'll dip out. Sometimes they'll just come and they'll sit and wait for someone else to ask a question. I have no problem with that. I'm not going to answer a question that's not asked, though. So if you come and join the meeting and no one else, or if you join the office hours and no one else is there and you're just going to wait, I'm fine with that. Chances are I will unintentionally pick my nose or something like that, and that will just be humiliating for me. So you'll get to capture that moment where they're like, "Oh my gosh, that guy he accidentally picked his nose." That's embarrassing for him, and it will be. Okay, so like I said though, if you need a different time, contact me. Um, and the best option is if you contact me and you say, hey, tomorrow at 2 p.m., can we meet? Well, chances are, yes, because that's when our class is. But if you say, hey, sometime tomorrow morning or Thursday morning is the best time for me to meet, I would like you to propose a specific time so that we don't have to volley back, oh, does 10 a.m. work? No, it doesn't. Okay, what about 9 a.m.? No, it doesn't. Okay, how about 11:30? No, okay, yeah, that works. Propose a time and chances are I'll be able to accommodate that. This is the only class that I'm teaching in summer session one. So I'm hoping that I can take care of everything in a very timely fashion. By that, I mean getting you your grades back, getting you feedback, and responding to your emails. Um, the weekends, typically I check my email more than I want to. But I will say, if even in a weekend, if I don't respond to your email within 24 hours, please email me again. Because what that means is that I open the email and I said, ooh, I better respond to that. And then I promptly don't. But I will try to get back to you as soon as possible. Don't feel like you're pestering me or anything like that. Um, so, okay, those things are taken care of. Next up, textbook. The textbook that we're going to be using is an open educational resource or an OER. It is titled Biochemistry Free for All. And this is published by Oregon State University. That link right there will take you to this page, which should be popping up. Nope, it didn't. There we go. It will take you to that page. And what you'll see there is a couple of different things. There's Fundamentals of Cell Biology, not this class. Biochemistry Free for All 1.3, that is our class. You can access the iPad version, download the PowerPoint files by topic, or download the entire textbook. Um, my biggest criticism of this textbook, which I'll go ahead and open the PDF. My biggest criticism of this textbook is that, and this is really the case for most open educational resources, is that they're well done, but it's almost like there wasn't the editor who came back and looked at everything and said, hey, we can get a higher resolution image for that. So there's some places where it looks like rookie ball. I like baseball, so I'll use baseball references. So it looks a little bit amateur hour. Um, but I don't want that to take away from the publication itself. Um, the second item that is a little bit of a criticism is this book is 3,648 pages. And don't think about that as like, oh my gosh, this is like every other textbook, but four times as large. Instead, it's like they decided, hey, let's go with size 74 font and let's put as many pictures on this as possible. They did. Man, that's not a beefing, but yeah, so here, like look at this one. Figure 1.4, you can see that the yellow's kind of like blotchy there. And we zoomed in quite a bit. Yeah, the color backgrounds, there's room for improvement. Um, but that's like aesthetics. That's not content. The content of this book is just like any other biochemistry textbook. So I figured that a free one would be a good way to go. If you want a hard-covered copy of this, I'm sorry, there's not a hard-covered copy. If you want to print it, well, it's 3,648 pages. I would try to go front and back. And I would try to get like I don't know, probably need like a 7-inch three-ring binder to put all that into a binder. I don't even know if they make binders that large, but there you go. I also like that I can print up like one page or something like that and make my doodles on that. So anyway, this is a book that I like. It's got everything that we're looking for, and they even have links in here. I have found a couple that the links are dead, but there will be like I think it was on the first page. Like here you go, second page, I guess. YouTube lectures by Kevin here and here, so you can click those and get Kevin to give you a little bit of a rundown of that. I don't know who Kevin is, one of the authors or one of the contributors. Okay, so that's our textbook. Course objective to use critical thinking skills as well as quantitative and qualitative reasoning to gain a greater understanding of biochemistry and solutions brought on by the study of biochemistry. Our course description provides a challenging introduction to biochemistry covering the structure and function of nucleic acids, proteins, lipids, and carbohydrates. Major metabolic pathways of carbohydrates and lipids are also going to be examined. This course is not intended for biochemistry majors. Heard that before. And you got the co-rec of Organic 2. Chances are well, I don't know if Organic 2 is being offered right now. That probably doesn't apply for anyone in this class. Now, with that in mind, grading of this class, and this is where I'm going to kind of get into some of the weeds of it. Um, and then after I do this, I'll take a pause and you guys can ask questions if you have them, before we get back to it. So our grading is going to be based on quiz, exams, a final, and homework. I tried to make this as simple and streamlined as possible. I've got your first two homework assignments up on Canvas. I labeled them problem sets 1 and 2. Um, those homeworks are due at 11:59 p.m. on June 3rd, 6th, 13th, 20th, and 25th. A little bit staggered from when your exam dates are. And the reason for that is I want you to kind of do the adequate work or do the work for this so that you can answer these questions and then I can post the correct versions or the correct answers so that you can look at those before you take your exam. So the first two problem sets are up. Those are due June 3rd and June 6th. So that's a week from today and then a week from Thursday, I think. That sounds right. That's the volume that I just clicked. Yep, a week from today and a week from Friday. Sorry. Um, your homeworks, and I'm going to switch my screen for this because grading is where I don't necessarily get weird, but it gets a little bit complicated. Um, and okay, so yeah, there we go. Yes, as I said, biochemistry 4375 is party time. Let's see, that's okay. Is my tablet working? Should be. I got the right board, don't I? Just a second. think that's wrong. This, are you doing this to my computer? One more attempt. Okay. Tablet. Okay, this should do it. And well, that's frustrating. Um, okay, so what we have is we've got homework, two problem sets are up. You're going to have a total of four of or five of them, and your lowest is going to be dropped. Um, these homework assignments can be accepted late. Oh, homework can be accepted late, um, but I believe that I've got it in the grade book as if it's one day late within 24 hours, it's 10% deduction. And then if it's 2 days, or it's within 2 days, it's 20% deduction. But after that, it's not accepted anymore. So make sure that you get your homework in on time. The quizzes, same sort of thing. I believe we got four of them, four or five of them. It says on the number of dates. I know somebody's going to chime in with, okay, we've got, yep, five. Five quizzes, lowest one is going to be dropped. If you miss one, well, then don't miss another one. Those keep in mind though, they're each of those categories are worth 10% of your grade. So in the event that you miss a quiz, you could, or miss two quizzes, we'll say, you could kind of view that as, okay, the highest that I can get in the class is probably about a 97.5%. So that's a solid A plus. So you can still maximize it. Where it gets a little bit hairy in my class is when it comes to exams. Um, I don't intend for this to be confusing or anything like that, uh, but it is a little bit. And the reason for that is because a um, I am using a kind of progressive and student-centered approach to grading. And that progressive and student-centered approach to grading is a couple of folks in the chemistry department have adopted it. Just a second. There we go. Um, it is cumulative grading. I'm not sure if anyone heard what I said because I'm really having technical frustrations right now. Cumulative grading. So, what this grading scheme is going to consist of is basically every exam is an opportunity to replace the previous exam. And so what that I mean by that is, and let me go ahead and change my screen, my display, so that I can show you the grade book and how it differs for me from you guys. Um, if you look at this right here, this is the way that I see your assignments. Um, and this is something that I think it would be helpful for you guys to see. Here's your quizzes. This is 10% of the total. Homework, 10% of the total. I'm going to apply a little rule for this. Your lowest one is going to be dropped. Okay. Now, how does that impact your exams? Well, when it comes to your exams, I've broken the class down into modules. Module 1, Module 2, Module 3. In module number one, you're going to take the first exam, which will be on June 9th, and you'll get a score for that exam. Then, whenever you take exam number two, which will be on June 16th, you'll get a score for that exam. Then the final exam is going to roll around, and that's on June 27th. What I'm going to do is combine exam number one, two, and your final into module number one, which is in the grade book effectively 15 or 20% of your grade. So these three items are going to be grouped together. Can I get a confirmation that yes, I follow that in the chat?
I think we're seeing something different than what you're seeing because what we see, you didn't circle anything just now.
That is slanderous, Aaron. I would never make a mistake that I just did. And I don't know if slander is the right word, but thank you very much for letting me know. Now, now, I believe you see the same thing as me. Okay. So we've got module number one. Think about that as a grade category that's 20% of your grade. In there will be the score that you got on exam number one, the score that you got on exam number two. Thank you, Mila. And the final exam. That's right. I think it's libel if it's written. Anyway, so those three grades will be grouped together. They'll be in one pool, and that pool is worth 20%. Now, check this out. That's three items. Here's the rule that's going to be applied. I'm going to drop the lowest two. So what that means is, let's say hypothetically that on exam number one, I'm going to type the numbers in here. Exam number one, let's say that you got a 95. Then you got a 95 on exam number one. Great job. Exam number two. Maybe what happens is you're riding high a little bit. And you're like, ha, I got this stuff. And you get a 75. And then the final exam. And this is, of course, at the end of the semester. You got a unit 87. What's going to happen there is the 75 that you got on exam number two and the 87 that you got for the final are not going to relate to this 20% of their grade. So now, just so I like to make this as clear as possible so that I can kind of revisit it. Let's go ahead and let's imagine now we're talking about module number three. You have your third exam, and let's say you got a 93 on that. The final exam, again, we got an 87. Here, I'm going to apply a rule where I drop only one. I'm only going to be comparing one thing. So I'm going to say lowest score, drop one lowest score. What the gradebook is going to do is it's going to ignore this final exam score. So with that in mind, one of the questions that people always like to ask in this instance is, well, do I have to take the final if I've made straight A's? And the answer to that is yes. If you aced exam one, two, and three, perfect. Great job. The final exam is in its own category, which is worth 20% of your grade. Now, one extreme way that I always like to think about this also is for the person who, let's say over the course of the semester, and maybe I can adjust this enough, I think. Yeah, there we go. I'm going to use this display so that I can populate with all sorts of different grades. Okay, exam number one, somebody got a 95. Then exam number two, they got let's say a 75. Then exam number three, you get a 93. And if these numbers are different from what I just said a second ago, I apologize. The final exam, you get a 97. Okay. What's going to happen is I'll go back and basically duplicate some of these grades. Exam 2 grade, that was a 75. Exam 2 score will be shown in two different locations in the gradebook. The final exam, that's where you got a 97, 97, 97. And then the last one is exam number 3 in module 2. And that's where you got a 93. So for this, well, when we look at basically the importance of the final exam, well, on the final exam, this person did better on the module 1 content than they previously had. Same thing for module 2. Same thing for module 3. And their final exam was worth that alone. So in this specific instance, what was the approximate weight of their final exam? And you can add this to the chat. What percentage of their grade was their final exam worth? 80%. Because that person did better on their final exam than all of their previous exams. Now, is that going to be the number that's the case for everyone? Not at all. So now I'm going to change these numbers a little bit. And let's say that we have someone who on the final exam, okay, sorry, just a second. Let's say somebody else on the final exam, they get a 45. In this case, I'm not going to circle all the numbers again. Approximately what is the weight of this person's final exam? Perfect. 20%. Did their final exam performance hurt their grade? It did. Did it bring down any of their previous exams? It did not. Now, a fair question for why am I doing this, or a fair answer to the question of why am I doing this? Well, the reason is because on the final exam, I'll put it in the context or through the lens of the final exam. On the final exam, are you going to be asked questions related to module number 1? Yes. On exam number, or sorry, what about module 2 and 3? Yes. The final exam is going to be cumulative. So you will see module 1, 2, and 3 stuff on there. So it's kind of like you're revisiting that material. Now, what that means is that when you take exam number 2, right here, that is going to have both module 2 and module 1 content. So this kind of introductory, the first 2 weeks of material is going to be on exam number 2. In addition to, well, the module 2 stuff. So what that means is that every successive exam has a little bit more content, but the idea is that you're reinforcing it. So what I mean by that is when you take exam number 2 on June 16th, you're going to have questions from module number 1, but you will also already have feedback from module number 1. So if there was something that you got tripped up on and you didn't answer correctly, a type of problem that you struggled with, you have the opportunity to go back and make sure that you know how to do it. So when exam number 2 rolls around, you're going to get that question right. Now, does anyone have any questions related to grading? Is everyone clear on, I guess just give me a thumbs up or some sort of response in the chat if you're clear on the fact that the final exam is mandatory. Well, not mandatory, nothing's mandatory, but it is worth 20%. Thumbs ups, celebration. Okay, great, great. Getting good responses there. Okay, cool. Let me clear all my drawings here. Um, all right. So that's okay, good, 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 good. The next item before we get into kind of going through material for the class is that every Zoom meeting is recorded, and I post the Zoom recordings to my YouTube channel and then send you guys that link, um, within a couple of hours after class meets. So they'll be available for you. If there's anything that you want to revisit, you're more than welcome to. I try to get the link up by our class is done at 3:40, so I'll try to have the link up by like 6 p.m. or so. Just because Zoom takes a little bit of time to process the video so that then I can publish it. So I'll get it to you as fast as I can though, so that you have that, and you can kind of scrub through it. And if there's any reason that you have to miss a class, then I'm not really taking attendance or anything like that. You're just not going to have the opportunity to ask a question live. You're more than welcome to ask a question later on, but that's just one of the issues. Then let's see. So I as you can see from our percentages here, I don't have any sort of participation or attendance expectation. Um, then let's see. So there's recordings will be posted. Yeah, and whenever I throw them up on YouTube, then I share the link to our Canvas page or share the link on our Canvas page. Um, and actually, let me before I get into things, I'll go ahead and jump back here. Um, usually, so I got the calendar, the homepage structured in this way. And usually at the bottom of one of these cells, for instance, down here, you see agenda, item, syllabus, textbook. There's going to be a link probably below that that just says, recorded lecture. So look for it there. Um, in the event that I forget to do that, I apologize in advance. If you don't see the link up there by like 8 p.m., email me and I'll say, oh, I'm sorry. I will get it taken care of. Usually between 6 and like 10 p.m., I don't respond to or do anything with emails because that's usually whenever my family's having dinner and I'm helping put my kids to bed. So if if I don't get something done by 6 p.m., I'm probably not going to do it till 10 p.m. So that's why I like to get things done before then. But if you don't see something, please don't hesitate to email me and say, "Hey, I can't find this," and I'll get back to you. And usually just full transparency, usually what happens is I'll get the email and then I'll say to my wife, like, my students can't find, oh my, oh, oh, oh. And then I'll look at the Canvas page and I'll see there's no link there and I'll be like, but still, they should have. Um, and then I'll usually respond with like, hey, sorry, here's the link. So I will filter that as opposed to being like, here's my angry email to you. Okay. Um, so here's the rundown of the class. If you've on the left-hand side, I've got the week's learning objectives. Um, and I want you guys to have access to these. I'm going to make them available as we visit them. So for instance, general and background information. That learning objective, that one is published, and these are kind of the learning objectives that I want you to walk away from this first section. Most of this we're going to cover today. There's a couple of things that we'll catch tomorrow and wrap up then. There's a couple of learning objectives that I have not yet published, and the reason for that is just because we haven't gotten into them yet. For instance, well, no, that one's good. Um, but amino acids, I think that one is not published, right, Joel? Yeah, good. So it's still a secret. Um, that's kind of if you look, however, at the week's schedule, and just so you guys understand my context, um, I do most everything on Canvas on a web browser. I don't use the Canvas app. So it's entirely possible that the display that you have is a little bit different from mine. In which case, sorry. Um, so I always go to the Canvas or the homepage of my class. This item, amino acids, protein and genetic amino acids are super important for the study of biochemistry. So we're actually probably going to get to them maybe as early as Thursday, just because there's a lot of, dare I say, memorization that's important for you guys to kind of be ready to tackle. So the earlier we get to it, the better. We'll be doing some things leading up to structures of amino acids that will, I think, be beneficial, so that then you know how they work and have a little bit of better understanding of that. Um, I know that if you click on syllabus on your Canvas page, you'll see the downloadable document as well as all of the dates for everything. Um, but I've also got I tried to have this calendar right here ready to go, so that you can see like, oh, it's Tuesday the 3rd. I've got a problem set due by 11:59 p.m. tonight. So I try to keep that up to date and clear so that you're good in that respect. I've tried to distribute everything pretty evenly throughout the semester so that it's not like, hey, you've got 6 homeworks due on one day. Because I want you guys to stay on pace with this. Um, I've tried to also make sure that the quizzes, which you'll see on Monday, the quizzes and problem sets are aligned with the learning objectives so that it's not just like you have learning objectives out here and then quiz questions that are completely unrelated to them. I want you guys to have like, oh, that's what where should you go to study? Where should you go to prepare for a quiz? Well, check out the learning objectives, then look back at the lecture and say, okay, did we address that? Is that something that I need to brush up on? Um, I tried to also make sure that the deadlines were 11:59 p.m. so that I could avoid any sort of ambiguity, in terms of like, no, it's not at midnight, just because I will always struggle with, do you mean midnight tonight or tomorrow? Um, so everything's due. For instance, quiz number one, that will open at 12 p.m. Yeah, 12 p.m. on June 2nd, and you'll have 11 hours and 59 minutes in which to complete it. I believe this is what you would see if you clicked on this quiz. Currently, it's worth 0 points because it doesn't have any questions in it. The answers will be shuffled. There's 20 minutes in which you have to take it. It's not going to be administered through Proctorio. You're going to have one attempt at it, and you got a time limit of 20 minutes. Um, and that's the quiz. Those are the quizzes. The homework or problem set number one, um, all of your questions are here. One thing that I want to clear up is, um, I your answers must be written using your own penmanship. I tried to clear up any sort of confusion there. Like you do not have to, it doesn't have to be written on a piece of paper. If you have a tablet or something, you're welcome to use that and draft it on there. Are they multiple choice? The quiz and exams are going to be multiple choice with a handful, not a large number, but a handful of multiple select questions. So it would be like select the true statements below sort of thing. I'll get more into those before our first quiz. Um, that's a important and good question though. Let's see. Yeah, so I want you to write this. Don't type it. If there's a prompt that says draw, I want you to draw it. I don't want you to find a picture of it and copy and paste it. Um, are you going to be expected to draw something on an exam or a quiz? No. Oh, so then why are we having to draw it here? Because the act of drawing something helps you remember it and kind of, I'll use YouTube language here, and it encodes it into your brain DNA. Brain DNA, yeah. But I think that it's beneficial to actually drawing it. You can identify, like when you draw out, oh yeah, I drew three bonds there, or I drew two bonds, a single, okay, and a lone pair of electrons. Okay. So this is the place where we're going to be doing those sorts of things. Um, Last thing about the calendar is um, on the 12th and 13th, our lectures are going to be pre-recorded. So I will have those available for you before those dates. If you have questions on those dates, you're more than welcome to ask me, but those are going to be two designated pre-recorded lectures. Um, and yeah, yeah, that will be after your first exam. And, this is the benefit, or sorry, the benefit of a online hybrid course is there's like time flexibility, I think. Um, but then, yeah, that's one benefit. So in the event that you need to meet with me and you're like, oh, can we meet on, Sunday afternoon? Yeah, I can probably carve out some time to do that. Um, this is a summer class, so it's going to go at a very high speed. And I know that I've spent 48 minutes talking about stuff that is not specifically biochemistry, but I think that it's important to cover all this stuff. Before I go back to the PowerPoint, does anyone have any questions about the structure of this class, where your grades are going to come from, anything like that? If you do not, oh, will you have to take online exams during class hours? It's a great question. For those of you who are going to take your exams online, those will be administered between 1:30 p.m. That's the earliest that you can start it, and I think 4:30. So, to answer the question of during class hours, if you're a hardliner, no. It's going to be very close to your class time. It will be slightly different though. So you can start a little bit earlier and yeah, until 4:30 p.m. And if this um yeah, so that's kind of the plan. Just because I always want to include a little bit of additional time if somebody's doing it online just to avoid technical issues and to be able to address technical issues. Okay. Well then I'm gonna go ahead and switch over. So clear that. Okay. So first things first when it comes to uh well, a living organism. If you've taken a biology class, if you've taken a microbiology class, this is going to be a little bit of a review for you. But what we're interested in biochemistry is prokaryotes and eukaryotes. There's a handful of different details about prokaryotes versus eukaryotes. For instance, well, prokaryotes versus eukaryotes. Eukaryotes do not harbor their DNA in a membrane-bound organelle, and membrane-bound organelles are maybe the most significant and important difference between the two different types of cells. Another big factor is that, well, a eukaryotic cell is about 10 times larger, at least, than a prokaryote. Now, when we look at a prokaryote, oat, I always say oat. A nucleoid, that's the irregularly shaped region where genomic DNA, as a circular chromosome, is how it's typically depicted, is found within a bacterium. Um, Genomic DNA, that's all your DNA that basically makes this organism go on to be the next generation of organisms. Um, Bacterial chromosomes are typically circular, not always, but they are typically circular. Most or many eukaryotic chromosomes are linear. There are some that are circular, and there are some, in the case of yeast, that can be, Well, they can be circular at some points and they can be linear at other times. Another structure important to a prokaryote is the pili or pilus. These are short hairs right here that help bacterial cells attach to things. In the case of a um, in the case of a bacteria that needs to attach to a host, the pilus or pili are going to assist in that. A cell wall is a structure that is unique to a prokaryote, or not completely unique to a prokaryote, but is important for a prokaryote. This structure provides protection and support beyond the plasma membrane. So when it comes to a prokaryote, we've got three major structures that separate them from a eukaryote. Or sorry, four, because there's also the flagellum. A flagellum is, I always kind of visualized it. I know that molecularly it's a little bit different from this, but it's a big hair that enables cellular mobility and motility. So it enables this bacterial cell to move around. Within a prokaryotic cell is the cytoplasm, the interior space of a cell where reactions are going to take place. And this is made up of a gel-like solution known as cytosol. A ribosome, these little blebs right here, are the sites of protein translation. The plasma membrane, it's the membrane or the protective outer layer of a cell that keeps the good stuff in and the bad stuff out. Typically is what it's going to do because it's going to be able to have a selective or have a mechanism to select certain things to keep them in and then to keep other things out. Now, in terms of the parts of a prokaryote that are important, well, flagellum, nucleoid, pilus, and the cell wall. Now, when we talk about a eukaryote, well, eukaryotes are larger than prokaryotes, and they've got a couple of structures that make them different. There are some structures that are similar. For instance, eukaryotes also have a plasma membrane. They also have ribosomes where protein translation takes place, and they also have a cytoplasm with cytosol, that gel-like solution inside of the cell. The structures that are unique for a eukaryote, and this is not all of them, this is not a completely comprehensive list, but these are some of the important ones. They have a Golgi apparatus. This is a structure that's primarily responsible for the modification, sorting, and packaging of proteins. Now, a fair question there is, well, packaging for what? Well, one thing is that those proteins might be exocytosed. What that means is that they're going to be packaged up and then delivered to the plasma membrane. That vesicle or that kind of package will merge with the plasma membrane, and that protein will be released to go outside of the cell. The nucleus is akin to the nucleoid in the sense that it's where genomic DNA is stored, but it's more defined because it's going to have a membrane, whereas the nucleoid does not. The endoplasmic reticulum, also known as the ER, there are two variations or two versions of this. There's the rough ER or rough endoplasmic reticulum, and then there's the smooth ER. These do different things. They modify proteins, and they're also involved in drug detoxification. Now, the last two, the lysosome is a structure that does lots of breakdown. So it can take large fats, it can take proteins and break them down into their individual parts. The very last structure on this list is the mitochondria, or the mitochondria when we're talking about plural or multiple of them. This is a structure that's commonly known as this is what I love, the powerhouse of the cell. There was at least one person in this class who I said the mitochondrion is also known as there's at least one person in this class who, through like muscle memory, did this. They mouthed powerhouse of the cell. So that's where ATP is generated, a large amount of ATP. Now, one thing that's changed over the past few years in this biochemistry class is we're going to talk a little bit about the mitochondria's role in ATP production. If you're not familiar with ATP, think about that as the energy currency of a cell. So ATP, or adenosine triphosphate, is a molecule that can be broken in order to drive lots of reactions. It's a high energy compound. It also happens to be a nucleotide triphosphate, or a nucleic acid. So when you hear DNA and you think deoxyribonucleic acid, well, ATP is a ribonucleic acid. We're going to get more into that later, but the mitochondria is where that a huge amount of that compound is produced that can then be released from the mitochondria to be used by, well, tons of different components in the cytoplasm. So the mitochondria, of all of the organelles that we're going to talk about in this class, we're going to lean most heavily on the mitochondria because that's where lots of energy is produced. And it's kind of a focal point of central metabolism. Now, if you are a biology or a chemistry major or a pre-health major of some sort, chances are you're going to take Chem 4385, which is metabolism. Um, that is a class where you're going to go deeper into what we're talking about here. So, what we will do in this class in terms of central metabolism or primary metabolism is we will talk about glycolysis, the process by which sugars are broken down to make ATP. We'll also talk about beta oxidation, which is the process by which lipids are broken down to make ATP. So we'll talk about those. In metabolism, you will spend time talking about those two, but then you'll go deeper and you'll talk about like photosynthesis, you'll talk about um, lipid synthesis, so making fats or making lipids. Um, in this class, I just want to make sure that you have some more context for metabolism than historically you did. In addition to that, this is just a little bit of an aside. If you are planning on taking the MCAT, um, and you're planning on going to medical school or maybe PA school or something like that, you've probably seen that the pre-health, like the pre-health curriculum says to take 1 semester of biochemistry. Um, and maybe they recommend two, I'm not sure exactly. But people take 1 semester of biochemistry and then they go to take the MCAT and they think, oh, I took 1 semester of biochemistry. I should be good. And then they get to the MCAT and they're like, oh my gosh, this is asking me a lot about metabolism. Um, and that's pretty standard across the United States. Like most universities, they have a 1 semester biochemistry class that it's like the organization that's responsible for drafting the MCAT doesn't really know what's taught in that class. No, that sounds like I'm that doesn't that sounds much more mean than I want it to. Anyway, the point is we're giving you kind of the tools to understand metabolism that you will then, when you take metabolism 4385, you'll get a deeper understanding of. So this is we've revised this in the past few years to make sure that you're in good shape should you take the MCAT after this class. Okay, so um, when it comes to domains of life and organisms, we've got three major branches. We've got bacteria, archaea, and eukarya. So we've got eukaryotes, and then primarily these two are going to be prokaryotes. This tree of life here shows a LUCA, or last unknown common ancestor. So when we look at animals and fungi, their common ancestor with plants is right about here. And then you can go back further and further and further and see, okay, well, this is where the branch of archaea split from eukarya. Okay, cool. Um, Archaea typically flourish in extreme environments. Commonly, you'll hear them referred to as extremophiles. And then one thing about all three of these different branches is that they can all have one thing of a cell wall. Um, Cell walls are more commonly found in your bacteria and archaea, but eukaryotes, like plants, can have cell walls. Now, this first lecture, we're kind of a little bit all over the place. Um, One thing that I want you to think back on with respect to your previous coursework is to think about organic chemistry. And in organic chemistry, you talked about functional groups. When you think about a functional group, um, what term comes to mind? Go ahead and add it to the chat. Or do any functional groups come to mind? Ketones, carbonyls, aldehydes, great. Okay. Now, with these different functional groups, whenever I see aldehydes and ketones, the first thing that comes to my mind is a partial charge. So partial charges of different on different groups, lone pairs of electrons. These are all important for biochemists. Now, when you think about, I saw somebody say a carbonyl group. If we were to draw this guy right here, and bear with me, my tablet for some reason is not working. Okay, fine. The reason is because I didn't test it a couple of days ago. Um, but what is that? Add to the chat what that kind of decently drawn group is. Carboxylic acid. Okay. Now think about a carboxylic acid. Is there anything that the way that I've drawn it, is that in an acidic, a neutral, or a basic environment? Add an A for acidic to the chat, a B for basic, or an N for neutral, the way that I've drawn it. Okay. Seeing lots of acidic. Perfect. Okay. Now, I think that you guys all know that because I'm thinking of a number that indicates the strength of that. And I'm thinking of three letters. Relate that number is also known as a pKa. Perfect. A pKa is something really important for biochemists because, if I were to look at this, any carboxylic acid, could anyone make an estimation about what the pKa of, kind of a general carboxylic acid is? I'm going to do the organic chemist thing and just throw an R here and done. That could be 6,000 carbons there, but make an estimation about what that is, that pKa. I got 35. I'm thinking a little lower than that. And maybe it would be helpful if I said acetic acid. Oh, about 5. I like that. So acetic acid's pKa, I think, is like 4.37, but let's go with 5. Okay, so a pKa of about 5. Now, if I establish that, yes, my pKa of this is about 5, not quite 1 million, but we're going with 5. Let's say that I threw this into a solution, threw this molecule into a solution with a pH of 9. A fairly basic pH, not extremely basic, but decently basic. Um, is the molecule that I have drawn and circled, is that going to change at all? Yeah. Okay. Now, follow-up question. Does my pKa of that molecule change at all? Add it a Y or an N to the chat. Does my pKa change? No, it does not. If I threw it into a solution with a pH of 10, the pKa would not change. If I threw it into a solution with a pK or a pH of 3, the pKa does not change because the pKa is something intrinsic to that molecule. So whenever we say, oh, the pKa of this group or of this molecule is 5.2, that's what it is. It responds to the environment, but that number, that pKa, does not change. So this molecule that I've got drawn here is going to change. How does it change? Well, what's going to happen is in a pH of 9, it's going to lose that proton and it's going to be negatively charged. When the pH is above my pKa, well, then that molecule is going to be deprotonated. So that right there is deprotonated, and it happens to have a negative charge. Understanding this is at the core to understanding a lot of what we're going to be doing with respect to amino acids. So if ever you've heard anything about a biochemistry class and you've heard, oh, you got to know your protein, you got to know those amino acid structures. Yes, you do. That's like half the battle. The other half of it is understanding how these molecules change based on the solution that they're in. Because that first question, what sort of environment do biochemical reactions take place in? Are they going to take place in something like hexane? Oh, I hope not. Butane? Oh, again, I hope not. They're not taking place in an organic environment. Biochemical reactions will take place in an aqueous environment. Something where the pH or the pH of the solution might be 7.2, might be 9. Functional groups in a aqueous environment are going to respond. They're going to change based on the pH. We're not doing this in an organic environment, which is one of the reasons that I think it's appropriate that you don't have to have organic 2 in order to excel in this class, because we're talking about stuff in a different environment. We're talking about something in an aqueous solution. So what we want to know is, how do these groups differ from one another? How do they respond to the environment? And how do they interact with one another? A negatively charged group, what is a negatively charged group going to be attracted to? Add to the chat. Perfect. A proton or just a positively charged group. You're absolutely right. So if you understand that, that's going to be a good thing for you as a biochemist. Okay. Now, groups change based on the pH. A carboxyl group is a good example. We just drew that. Now, complexity. In this class, what we're going to be doing is we're going to be talking a lot about polymers. And we're not going to be talking about polymers in the sense of like making styrofoam or dental floss or anything like that. Um, but what I want you to take away from this is we're going to be talking about individual atoms as well as small molecules and how they grow into something at the macromolecular level. For instance, we'll talk about individual sugars, and we'll talk about sugars as polymers. We'll talk about glucose, and we'll talk about glycogen. Nucleic acids. Well, we'll talk about adenine, the nitrogenous base represented by the single letter code A, and we'll also talk about genome stability. We'll talk about proteins. That's your macromolecular structure. And we'll talk about amino acids that build up to make a protein. The last major biological macromolecule or class of biological macromolecules are lipids. Lipids do not actually assemble into polymers, unlike sugars, protein, or sugars, amino acids, and nucleic acids. Lipids are going to, they'll form like sorry, if you've ever heard of a triacyl glycerol or a triglyceride, well, that's kind of the piece that has three fatty acids or three lipids and glycerol. So three fatty acids plus glycerol will make a triacylglycerol. And those are major parts of membranes, but that's not going to make a polymer. So I want you to keep this in mind in terms of polymers that you can expect from biological macromolecules. Um, yeah, so there we go. Then, just as a little bit of an aside, um, for the first time in my life, I've read, I'm finishing up the book Jurassic Park, and I don't know if anyone's ever seen that movie. If you haven't, I think that you should watch it because it's my favorite movie of all time. Um, but what I found kind of interesting about the book is that they not once use the word genome, despite it being all about, making, or I have not seen it. It's all kind of like, oh yeah, DNA manipulation, DNA manipulation. Yeah, we build it like this, but yeah, a little bit of a limitation. So if you're planning on using that book to construct a dinosaur, it's going to leave some gaps because I was planning on it. Okay, so we'll also discuss organisms and their fuel sources. This is something that you'll get into a little bit more in metabolism because you'll talk about photosynthetic organisms. Um, but we'll talk about uh, yeah, different organisms that use oxygen in an organic compound to break it down into carbon dioxide and water. Animals do that. Fungi and bacteria are going to do that. Those are your heterotrophs, and then plants, algae, and many different bacteria are autotrophs, where they're going to use carbon dioxide to synthesize organic compounds, which they can then subsequently break down to make water and carbon dioxide. So, or yeah. Um, then this is where it's an important thing to remember. Thermodynamics. Thermodynamics are a part of the study of biochemistry. Can help us predict if a process is going to be, if it's possible, or if it's spontaneous or non-spontaneous. And ultimately, we're going to classify reactions as being favorable or non-favorable. So a biochemist's view of thermodynamics is that life obeys the laws of thermodynamics. Living organisms require energy. Biochemists study the processes by which energy is extracted, channeled, and consumed by living organisms. We study the relationship between the structure and function of biological macromolecules. We also study metabolism, how sugars are broken down, how fats are broken down, how much energy can be extracted from something. Thermodynamics or bioenergetics gives us a way to describe or characterize energy changes in any biochemical reaction. Now, for thermodynamics, um, it's concerned with only the initial and final energy states of a system or reaction components. It's not concerned with the mechanism or the process. The thermodynamics is not concerned with the electrons being pushed around or anything like that. The key points for thermodynamics are enthalpy, entropy, and free energy. So enthalpy, represented by delta H, represents the amount of heat absorbed or released by a reaction. Entropy, or a delta S, the change in entropy, represents the degree or order of randomness of a system. And delta G, or free energy, represents the amount of useful work that can be done by a chemical reaction. Enthalpy. What happens during a chemical reaction? Well, enthalpy tells us a lot. First of all, old bonds are broken, new bonds are formed. Energy is either consumed or released. An exothermic reaction is where delta H is less than zero. Heat is evolved by the system, and new bonds or newly formed bonds are more stable than their predecessors. Endothermic reactions. Heat is absorbed by the system, and new bonds are less stable than their predecessors. Entropy. represented by S or delta S. It's the entropy is a measure of disorder. Now, the second law of thermodynamics says that for any process, the entropy of the system and its surroundings always increases. An ordered state is low entropy. A disordered state is considered high entropy. Now, a change in entropy that has a negative value. So I've always had a good way, or I've always felt like I had a decent understanding of entropy in like a conceptual, or I thought that I had a good conceptual understanding of changes of entropy, but quantifying is a little bit more difficult. A delta S that is negative is observed when the final state is more ordered than the initial state, and your products are more complex and more ordered. So negative change in entropy, a delta S, means that you've gone to become, or something has become more ordered. A positive delta S value is when your products are less complex and more disordered. This is observed when the final state is less ordered than the initial state. Free energy change, delta G, that's where we're going to primarily be. The reason for that is because lots of what we'll talk about in this class, when it comes to biochemical reactions, we'll look at what's the delta G, what's the change in free energy. And we'll be able to look at it and say, well, is that reaction spontaneous or non-spontaneous? And we'll describe a reaction as being exergonic or endergonic. Now, the change in free energy is used to determine the spontaneity of a process. Delta G equals delta H minus T delta S, the Gibbs free energy equation, which you probably recall from Gen Chem 2. You know, kind of, if you took that class in the spring, basically the time of the class after spring break, you probably got into the Gibbs free energy equation. Delta G, a negative delta G value, when it's less than 0, is considered exergonic. This indicates a spontaneous reaction and energy is released. When delta G is greater than 0, it's an endergonic reaction. This is a non-spontaneous reaction and energy is required in order to drive it. Okay, so when we look at biochemical reactions, delta G is at less than or greater than 0. That's the good question. That's the big question to ask. Now, when you look at the Gibbs free energy equation, well, that brings into account the heat of the reaction, the temperature, T, at which it takes place, and the change in entropy. When you look at those differences, and you can come up with delta G or a free energy change, it gives you a sense of, well, is this reaction going to take place when we account for everything? So when it takes place at 30 degrees or at 270 degrees, that's accounted for when you're given a delta G value. Now, one important thing about the Gibbs free energy equation, which I think that I have, yeah, here it is. This is the only measure of if a reaction is spontaneous or non-spontaneous. Delta H and delta S alone are insufficient to determine if a reaction is spontaneous. Now, if you look at this plot right here, this graph right here, what I want you to see is essentially this has a midline of 0, positive values and negative values. What this looks at is, well, if the delta H is positive and the product of negative T delta S is positive, what is that going to mean for delta G? Well, that is going to be positive as well. That's how we read this display right here. So this is taking into account, well, what are the implications of if delta H is positive or delta H is negative? I'll bring your attention down to this part right here with my poorly drawn star. If delta H is negative, what does that mean about delta G? Well, that alone isn't going to answer that question. But if you can say, well, the delta S or negative T delta S is also negative, well, then that means our delta G is going to be negative. Therefore, we're going to have a spontaneous exergonic reaction. Can I get some response in the chat for I follow, I don't follow? I'll go with a thumbs up for I follow, and then, or actually a number for I follow, a letter for no, I don't follow. Okay. So to rephrase this, this is what we care about. Delta G. We care about, well, is a reaction going to be spontaneous or non-spontaneous? We have delta H that says, well, heat is absorbed or heat is released. We have delta S that says, did this system become more or less ordered? So, and then T here is another part of this equation. Well, what's the temperature at which this reaction took place? When we have all of those variables, delta H, T, and delta S taken into consideration, we can come up with or conclude that the free energy change of this reaction is negative. Therefore, our reaction is going to be spontaneous. If delta H, if the value for this is 50, yep, that's a 50. If that is 50, that's a positive value. I'm going to get my eraser. What does that mean for our delta G value? Well, if delta H is positive, looking at that alone, that would mean that our reaction is going to be also positive, or our delta G is going to be positive. But what we need to say is, well, if the sum or if the product of T and delta S is 100, well, then that's 50 minus 100 gives us negative 50. So this equation and this graph over here, what it is all about is reconciling or looking at how positive it is or how negative it is. That's the point of what this is displaying, where you're trying to look at, okay, is this reaction going to be favorable from from the perspective of enthalpy? No, it's not. But from the perspective of the entropy change, yes, it is. So that's what we're trying to display in this graph. Now, if you want some more clarification or want to discuss this a little bit more, I'm glad to do that. But the point of this is if I were to give you numbers and you were to plug this in, I'd want you to be able to come up with, okay, well, delta G is positive. Therefore, it's a non-spontaneous reaction. If delta G is negative 37, well, that's a spontaneous reaction. I bring these up because when we look at different chemical reactions, that's what we're looking to answer. Is this reaction spontaneous or non-spontaneous from the vantage point of delta G? Now, when we think about chemical reactions, it's also very important to think about equilibrium. We've got a definition for equilibrium. The rate of a forward reaction is the same as the rate of the reverse reaction, and there's no net change in concentrations of reactants and products. Now, if you think about equilibrium, and I know whenever I think about equilibrium, I always go back to ice tables. We're not going to have to do any sort of ice tables. The initial change, equilibrium. And then I also think about Q, your reaction quotient, where you talked about, well, at equilibrium, here's the concentration, and here's the non-equilibrium conditions. We're not going to deal with that, but it's still relevant and important. Because when we talked in Gen Chem 2 about equilibrium, we talked about standard conditions. if you remember standard conditions, that was the standard state. You learned about these standard conditions where you said, okay, well, when you started a reaction with 1 molar of each of the reactants and 1 molar of each of the products, um, the reaction went towards equilibrium, and here's what it achieved. Well, one of the things with standard states and biochemists is that as a biochemist, we use, or one of our reactant is a proton. If you had, add to the chat if you know anything about what would the, can anyone describe a solution with, that's an H plus, concentration of 1 molar? Can anyone say anything about a solution with a hydrogen ion concentration of 1 molar? I mean, you could say that's a lot of protons. It is. What are the implications of a solution, or what do you know about a solution with a lot of protons in it? Super acidic. The pH would basically be 0. Okay. Um, I don't know about you guys, but within any of your cells in your body or any living system you've ever heard of, how many of them have had a cellular pH of 0? I'm looking close to 0. Because if in any cell in your body, the pH was 0, that'd be far, far, far too acidic. Does everyone follow me there? Okay. So my point is, chemists, and I'm not saying this to vilify or anything like that, or to draw a line in the sand or anything like that, but chemists use that standard state, and it makes sense. It's appropriate. But that standard state, where you might have a reaction with a 1 molar hydrogen ion concentration, that's not relevant biologically. Does that make sense? Because in any biological system, you're never going to have a pH of 0. So a chemist standard state is a little bit different from a biochemist standard state. And we have to kind of modify those. Now, this is kind of the detail of all of that. We're talking about concentrations of reactants and products being 1 molar. Hydrogen ion concentration indicates your solution's pH. Well, hydrogen ion concentration of 1 means your pH is 0. Will not sustain life. The pH in most cells is close to 7. Maybe it's 7.2. In some membrane-bound organelles, it's going to be different from that. But the point of this is, whenever a biochemist is trying to establish like a standard, they can't use that. So instead, what they do is they just say, okay, well, 1 is used in place of hydrogen ion concentration. So you don't do the calculation where you say, oh, the hydrogen ion concentration is 1 or is equal to 1. In addition to that, biochemical reactions are buffered. Biological systems have buffers. So if you've ever heard the phrase, the blood buffer system, that's why the pH of your blood isn't going to dramatically change. If it did, that could be fatal. Additionally, in biochemical reactions, the concentration of water is extremely high, 55.5 molar. So in place of the concentration of water as a reactant or product, we just use the number 1 for that. Now, with that in mind, we modified, or biochemists modify the standard state to reflect these changes, and we use this symbol, delta G naught, NaUGHT prime. So anytime you see this, delta G not prime. Usually you won't see it with such poor handwriting. When you see that, you should think, oh, this is a biochemist's standard state. Now, delta G naught prime takes into account pH and is therefore more relevant to biological reactions. We as biochemists, however, will use both delta G naught, which is the chemist standard state, and delta G naught prime as reference points for comparisons. What we want to do is be able to figure out the free energy change of reaction. Delta G is what determines if a reaction is spontaneous. So if we need to look at it, well, we've got Q, which is the non-equilibrium conditions. This is more commonly known as your reaction quotient. Now, quite a bit of this stuff is just a reminder for establishing context. Like, hey, you maybe haven't thought about this in at least this semester, maybe two. This is all pertinent and relevant to what we're doing. This gives us a way to look at equilibrium constants, KEQs. At equilibrium, delta G is 0, and Q is equal to KEQ. Delta G naught prime is equal to negative RT times the natural log of KEQ. When KEQ is greater than 1, delta G naught prime is less than 0. That's a reaction that is going to favor your products. KEQ less than 1, well, delta G naught prime is going to be greater than 0 and therefore favor your reactants. If there's an important thing for you to remember, it would be all of this right here. What does KEQ tell me in terms of favoring products or reactants, and what does it tell me in terms of delta G naught? All of that to say, what we've been talking about, and we've got 7 minutes left, and that's the perfect amount of time. This right here is an important concept for biochemists, reaction coupling. There are many times that we will refer to a process or reactions. Well, the reason for that is this phenomenon, reaction coupling. This is where multiple reactions take place as a part of a process. What I want you to see right here is we've got a total of number 1, 2, 3 different reactions. Reaction number 1. Okay. To make sense of this plot, this is our delta G sub 1. This is our free energy change of this reaction. This reaction consists of the sugar glucose. And could anyone add to the chat what PI stands for? Inorganic phosphate. Perfect. Or phosphate's fine, just as well. So what this reaction number 1 shows the 6-carbon sugar glucose adding 1 phosphate to make glucose 6-phosphate. The question is that spontaneous? Now, if we look at this chart, on the y-axis, we've got 0 and infinity as our range. That delta G sub 1, that represents a positive value because it's going from a low number to a high number. So that is a positive delta G. Therefore, is reaction number 1 spontaneous or non-spontaneous? Non. Perfect. It's a non-spontaneous reaction. Can I get a number if yes, I follow, or a letter if no, I don't follow that? Okay, great. And just so you guys are familiar and comfortable with this, that's kind of the way that I like to communicate with groups in classes, just like, hey, response, kind of a binary question, binary response. So it looks like everyone feels good about that. Going from a low to a high number, we've got a non-spontaneous reaction. Reaction number 2 is that a spontaneous or a non-spontaneous reaction based on a comparison to reaction number one? We got color coding, so that kind of helps a little bit. Yeah, it's a spontaneous reaction. What this reaction consists of is ATP, a high energy compound, adenosine triphosphate, being broken into two components, adenosine diphosphate and a free phosphate. Okay, so the third reaction among these is glucose and ATP to make glucose 6 phosphate and ADP. What we're trying to see here is that if you think about, let's say, it's a little bit like Hess's law from Gen Chem 1, where you take um, reactants from reaction number one, reactants from reaction number two. Reactants from reaction number one are glucose, inorganic phosphate, and now ATP whenever I look at reaction number two. ATP. Okay. Then my products from reaction number one, glucose 6 phosphate, G6P, plus ADP plus PI. What I want you to see here is that, well, inorganic phosphate is present on both the reactant and the product side. So I'm going to scribble that out. Now my reactants from my combined reaction are glucose and ATP. My products are glucose 6 phosphate and ADP. What I've done is I've combined these two reactions. One spontaneous, one non-spontaneous. What a biological system is capable of doing is taking that one spontaneous reaction and harnessing the energy from that spontaneous reaction to drive a non-spontaneous reaction, which is what we get with reaction number three. Does everyone follow that? A number for yes and a letter for I don't follow. Okay. So this is at the heart, or this is what is at the heart of lots and lots of biochemistry, and in particular, um, reactions within a metabolic pathway, where you have, maybe you have 10 steps. 3 of them might be unfavorable, but the other 7 are very favorable. 3 of them are non-spontaneous. Maybe they're super non-spontaneous, but the remaining 7 are very spontaneous. And so you're going to harness the energy to make those other reactions take place. Okay, it is 3:40 exactly. We might not always go up to 3:40 exactly, but today we are. I will be back tomorrow at 2 p.m. If you've got questions, I'm going to stop my recording and stop my share. I'm glad to hang around and answer any questions that you guys might have. But if it's easier or preferred, we can get into the Zoom office hour tomorrow. Just send me an email and we can set up a time. Other than that, I hope you guys have a wonderful day. Thank you for coming to class. Thanks for your engagement and responses and everything like that. And we'll keep that going. Deal? All right. Recording